Hi, today we're going to be talking about vegetable consumption and its impact on gastrointestinal health. My research question for this project was: does vegetable consumption promote gastrointestinal health? First, let's discuss the nutrients in vegetables. Vegetables are mainly composed of carbohydrates but also contain a variety of vitamins, including vitamin A, E, and C, as well as a variety of minerals, including calcium, iron, folate, potassium, magnesium, and zinc. They also contain antioxidants and dietary fiber. But we're mainly going to be focusing on dietary fiber. So, what exactly is dietary fiber? Dietary fiber describes carbohydrates that cannot be digested or absorbed by enzymes in the gut. So, there are two types of dietary fiber. The first type is soluble fiber, which can be digested and broken down by the body. It's not essential but beneficial to health, primarily gastrointestinal health and digestive health. Some examples of soluble fiber include pectin, beta glucan, and a variety of gums. The other type of fiber is insoluble fiber, which cannot be broken down by the body and passes quickly through the digestive system. Some examples of insoluble fiber include cellulose, hemicellulose, and lignin. This is an image that portrays the structures of both soluble fibers and insoluble fibers, specifically in the plant cell wall. Some vegetables that are high in dietary fiber include beans, like lima beans, mung beans, pinto beans, peas, like green peas, lentils, sugar snap peas, root vegetables, like sweet potato, carrots, and beets, cruciferous vegetables, like broccoli, Brussels sprouts, and kale, greens, such as spinach, artichoke, and Swiss chard, and squash, like pumpkin, butternut squash, and winter squash. As you can see, these are just a few examples, but many vegetables are high in dietary fiber. These are just some more examples, like navy beans have 16 grams of dietary fiber, green peas have 5.5 grams, broccoli 2.6, sweet potato 4 grams, butternut squash 3.2 grams, and artichoke 5 grams. So how exactly does dietary fiber consumption impact the body? We're mainly going to be focusing on the gut microbiota. The gut microbiota is a complex microbial community located primarily in the large intestine of humans, specifically in the colon. Some significant gut microbial species include firmicutes, bacteriotes, and proteobacteria. Some of these gut microbial species are beneficial to gut health, and some are less beneficial and can even be harmful, such as E. coli and Salmonella. But they are both important to keep in mind. We're going to be focusing on gut dysbiosis. So, what exactly is gut dysbiosis? It's a term referring to an imbalance of the microbes in the gut that can lead to a variety of diseases, including chronic diseases and inflammatory conditions. It can be caused by an overgrowth of bacteria or a lack of beneficial bacteria. In general, a balance of microbial diversity in the human gut is necessary to maintain the digestive system and prevent disease. Some conditions caused by gut dysbiosis include irritable bowel disease, which is a chronic intestinal disease. It can take place of Crohn's disease or colitis, which is inflammation of the large intestine. General inflammation is also caused by gut dysbiosis, which is just an inflammation of the digestive intestines, mainly the large intestine or the colon, or the tissues included in these areas. Colorectal cancer is cancer of the large intestine. The formation can be linked to altered gut microbiota composition or gut dysbiosis, and irritable bowel syndrome, which is also a chronic digestive condition associated with an imbalance of the gut microbiota. So, as you can see, a healthy balance of the gut microbiota is very significant to the health of your intestines and your body. So, how exactly is the gut microbiome controlled if it's so important? Certain types of foods directly control the microbial position by influencing the growth and depletion of certain bacterial species in the gut. This is a figure that very well represents a beneficial diet versus a typical Western diet high in sugar and high in fat that leads to mucus degradation, as well as reduced antimicrobial peptides that can lead to a pro-inflammatory immune response in the gut, and overall can lead to a dysfunctional gut barrier, inflammation, and even chronic disease, as previously mentioned. The first study that supports these findings is an animal clinical study. The goal of this study is aimed to assess the impact of fruit and vegetable consumption on gut microbiota composition. The method of the study used 46-week-old mice that were fed diets with different concentrations of fruit and vegetables for 15 days. Body weight and food intake were recorded and its effect on blood glucose, weight gain, and cecal content. The results of this study concluded that dietary intake of fruits and vegetables increased fermentation in the cecum and positively correlated with bacteria presence, specifically bacteriodes. The second study that supports these findings was a human clinical study. And the goal of this study was to examine the extent that whole grains and fruit and vegetable intake reduce the risk of metabolic disease by seeing the impact of fruit and vegetable consumption on gut microbiota and inflammatory markers. The method of this study was a random parallel feeding trial where 49 obese or overweight individuals with regular low whole grains, fruits, and vegetable intake were separated into 3 categories for high intake of each. Inflammatory markers and gut microbiota composition was measured over time. The results of the study concluded that there was a significant decrease in inflammatory markers with the increased fruits and vegetable diet, such as lipopolysaccharide binding protein and tumor necrosis factor. This figure displays the changes in inflammatory markers, and if you see A and B, the changes in lipopolysaccharide binding protein, as well as the change in tumor necrosis factor, there was significantly decreased with fruit and vegetable intake, which indicates less inflammation. There was also correlations between change in plasma markers and microbiota composition. Fruit and vegetable consumption decreased lipopolysaccharide binding protein and interleukin 6 inflammatory markers, as well as increased microbiota diversity, as seen in this graph on the bottom right. The third study that supports these findings was another animal clinical study, and the goal of this study assessed the effectiveness of supplementing dietary fiber in order to prevent and relieve irritable bowel disease by altering gut microbiota. The method of this study used mice induced with colitis that were fed barley leaf supplement diet for 28 days, and acute colitis symptoms were monitored. Colon tissue PCR and short-chain fatty acid and bile assessments were performed. The results of this study concluded that dietary fiber consumption can elevate abundance of beneficial gut bacteria, as well as decrease pro-inflammatory cytokines and have an anti-colitis effect. Overall, dietary fiber correlates with decreased colon inflammation and less severe bowel disease. The first figure for this study was a correlation between dietary fiber and colitis symptoms. As you can see in the colon length graph, the DSS was the induced colitis, which was significantly shorter when the colitis was induced, but then when the barley leaf was introduced into the diet, the colon was elongated, which indicates a healthier colon. And then the pathological score with the induced colitis was significantly high, but with the barley leaf, it decreased, which signifies that there was less colitis symptoms. The histology slides at the bottom with the induced colitis, the cells, the histology cells of the colon are dead. They're not purple anymore, less blood flow. But with the barley leaf, the colon is almost, so it looks as if it's almost returned back to normal, and the tissue looks healthy, which indicates a healthier colon. When looking at the dietary fiber and cytokine levels for this study, If you look at the serum cytokine levels and the tissue cytokine levels, they were significantly decreased when the barley leaf was introduced after the induced colitis had occurred, which significantly decreased these cytokine levels and also indicates less inflammatory response. Lastly, the dietary fiber and bacterial diversity graphs, especially from mice A, B, and C, indicate that there was an increase in fecal bacterial diversity after the barley leaf was introduced, which concludes that the introduction of the barley leaf allowed the beneficial bacteria to grow and helped regulate the gut microbiota. The overall impact of vegetable intake on gastrointestinal health, um, it first modulates microbial diversity as seen in the studies. It decreases inflammation. It decreases colitis severity. It promotes successful fermentation and digestion. It reduces symptoms of gastrointestinal disease and reduces the risk of colorectal cancer, all by maintaining a beneficial gut microbiota and reducing inflammation overall. In conclusion, vegetables contain beneficial dietary fibers important to maintain gastrointestinal health. They promote a healthy digestive tract, and regular consumption promotes a healthy gut microbiota and prevention of gut dysbiosis and gastrointestinal diseases. Thank you. Remember to eat your vegetables.